In this section, we will discuss the cell-mediated arm of adaptive immunity.
In this next section, we will cover the branch of adaptive immunity known as cell-mediated immunity. Cell-mediated immunity is involved specifically in protection against intracellular pathogens, intracellular bacteria, viruses, and also cancer and tumors. Cell-mediated immunity is mostly associated with T cells, NK cells, and activated phagocytes. Kind of an overview of how this branch works. We have naive T cells that are activated in the lymph nodes by dendritic cells or other APCs presenting the cognate antigen. Remember that the T cells have to see this antigen or this peptide presented in the context of MHC in order to respond. If it's specific and if they have the right MHC, then they become mature T cells and they will go enter the circulation. The APCs will then present the same antigen to the T cells in the periphery, and at that point we get clonal expansion of effector cells, either via MHC class 1, which is enhancing and activating CD8 CTLs, which would lead to direct cytotoxicity of cells, or via MHC class 2, which will activate CD4 T helper cells and lead to cytokine secretion. Once we have activation and expansion, the source of antigen is eliminated, and the effector T cells will die. The memory T cells that are created will survive. Now, starting from the very beginning, remember that these immature T cells are kind of born through the pluripotent stem cells, the hematopoietic stem cells in the bone marrow. Before they mature, they leave the bone marrow as immature T cells and migrate into the thymus. Now, the thymus secretes thymopoietins 1 and 2 to promote differentiation of lymphocytes into T cells. Also, it's important to note that this thymus atrophies after puberty. So babies, if you look at their chest x-rays, you see something that's going on above the heart, a big shadow. That's the thymus. However, as they get older and as they progress to the point of puberty, this thymus goes shrinking smaller and smaller and smaller. Even though it shrinks, it can still direct T cell genesis in the lymph nodes. So while these T cells are in the thymus, their maturation will begin in the outer cortex. In the outer cortex, they differentiate to express CD3 and both CD4 and CD8. And at this point, they're known as double positive thymocytes. So these initial immature T cells entering the thymus will begin to express CD3, CD4, and CD8. Then they will acquire specific T cell receptors and commit to single antigen specificity. Like the B cells, these cells will make T cell receptors that will allow them to focus in on a single specific antigen. The TCR generation is also very similar to what we see in the B cells. Note that we have the alpha chain and the beta chain, similar to the light chain and the heavy chain. If you look at the way the gene rearrangement works, in fact, it's exactly the same. So here on the top of the slide, we have the alpha chain. We see that we have a region of V genes and a region of J genes. And just like in the light chain of the antibody, we're going to grab a random V and a random J, and we're going to stick them together. And then we're going to loop out everything else that's in between them so that we have them right next to each other, the V and the J gene that we've chosen. Then we're going to take that recombination, which forms the variable loop on our T cell receptor, and we're going to stick it to the C gene, which forms the constant, or the C loop on our T cell receptor. And we're going to loop out all of the J's that are in the middle, so that at the end we have the V and the J recombination, which makes up our specificity, and the C gene, which gives us structure. That's your alpha chain. Looking down at the bottom of the slide, looks very similar to the heavy chain synthesis, right? We have V genes, we have D genes, we have J genes, and we have a C gene. And the function is the same. The expression is the same. The whole pattern is the same. We take a D and we take a J, we stick them next to each other, loop out everything that's in the middle. Then we take a V and we stick it to the other side of the D, loop out everything that's in between those two. Now we have a V, a D, and a J right next to each other. That makes up the variable loop on the beta chain of the T cell receptor. Then we take that recombination, the VDJ recombination, and we move it into close proximity with the C, looping out all of the J's that are in between. And now we have the V, D, J making up our variable loop, and the C, which makes up our constant loop. VDJ giving us specificity, C giving us structure. Now we can see the macromolecular structure of the T cell receptor represented here. There we have our alpha chain and our beta chain. Remember that alpha chain had the VJ recombination. That makes up that V alpha loop. And the C alpha loop comes from that C gene. That's the alpha chain. And then the beta chain, we have the VDJ recombination, making up that V beta loop. And then we have the C, which is made up of the C gene. And so we have a heterodimer, alpha chain and beta chain, with a peptide binding cleft being right there where the alpha chain and the beta chain come together in the variable loops. So in addition to gaining a TCR, we also have to test this TCR. We have to educate the T cells in order to make sure that they respond appropriately. This happens in the medulla of the thymus. And the process goes so that the TCRs are exposed to MHC molecules loaded with self-peptides. TCRs that bind strongly are self-reactive and receive apoptotic signals. This is called negative selection. TCRs that bind with low avidity receive growth signals, positive selection. TCRs that don't bind at all die from the lack of signals. So again, through positive and negative selection, which we've discussed earlier, with the positive selection, we weed out the cells that don't bind at all. We want those TCRs that bind with low avidity. The TCRs that don't bind at all die from neglect. The TCRs that are binding too strongly will receive apoptotic signals because we don't want the T cells binding too strongly to the MHC. That could lead to autoimmunity. We want those middle-of-the-road T cells, low avidity. Those are the ones that survive. Those are the ones that receive the growth signals. Those are the ones that are educated, so to speak, and graduate thymic education. So as the double positive thymocytes, remember at this point they're still expressing CD4 and CD8. As they mature in the medulla and are undergoing this positive and negative selection, some TCRs will bind with higher affinity to MHC1 and others to MHC2. Those that bind with higher affinity to MHC class 1 will mature into CD8 T cells. Those that bind with a higher affinity to MHC class 2 will mature into CD4 T cells. Once they've matured, now we've tested them, we've made sure that they're the middle-of-the-road T cell receptors, and we've decided which ones they like better, MHC1 or MHC2, and so we've determined whether they're going to be CD8s or CD4s. Then they can now leave the thymus as naive T cells and migrate to lymph nodes. So here we have the CD8-3 TCR complex, the fully made, put together, naive T cell complex. On the left we see the CTL. We have the TCR, the alpha and beta chains, which are again responsible for seeking out that peptide and making sure that it's specific, it's the right peptide. We also see the CD8 being expressed, which is going to look at the MHC class 1, make sure that it's looking at the right type of cell. And then the CD3 we see also in conjunction. CD3 is involved with downstream intracellular activation of the T cell. On the right we have the T helper cell. Again, we have that TCR, the alpha and beta, that give the peptide specificity. We have the CD4 this time instead of the CD8, which is looking for that MHC class 2. And then again the CD3 group as well, which is involved again in downstream signaling and activation of the T cell. So a question, a 23-year-old woman develops Hashimoto's thyroiditis. Given the role of T cell activation in the pathogenesis of the disease, what organ may have malfunctioned during infancy? Is it A, thyroid, B, thymus, C, lymph nodes, D, bone marrow, E, spleen, or F, liver? The answer is B, thymus. T cell activation has a problem. What organ may have malfunctioned? You're going to think thymus. Now, the T cells can't function in and of themselves like the B cells can. They can't recognize antigen in its native conformation. They need help from these APCs. So as we've mentioned, the professional APCs, the APCs that go around activating these T cells, are dendritic cells, macrophages, and B cells. Some of the highlights, some of the things that you might want to know about some of these cells. Dendritic cells are considered the most effective APC at stimulating naive T cells. Why are they the most effective? Well, they express the highest level of co-stimulation molecules, and they express also the highest levels of MHC, class 2. Dendritic cells are located just under the skin and in the mucosa of most organs. Those that are located just under the skin are referred to as what? Langerhans cells, right? These guys can capture antigen and concentrate it in lymph nodes for presentation. So they can sit in the lymph nodes, they can sit in the mucosa. They sift through, remember, pinocytosis. They sift through filtrate, grab the antigen, and then they can concentrate it and then present it to T cells within the lymph nodes. Macrophages activate effector T cells out in the periphery. Remember, macrophages like to live out in the tissues, so they're involved with that secondary activation. After the T cells have been activated in the lymph nodes and they move out to the periphery, then they're reactivated by these macrophages. Another question. A 10-month-old infant has had 8 bacterial infections since birth. He has a family history of MHC class II gene mutations. Which class of cells is functioning normally? Is it A, B cells, B, dendritic cells, C, macrophages, D, Langerhans cells, or E, T cells? The answer here is E, T cells, which may be a little bit confusing at first, but let's talk about this and why this is the correct answer. It's saying that this 10-month-old infant has a family history of MHC class II gene mutations. So out of the cells, out of the choices that we are given, which one of these cells does not require, does not use MHC class II? Well, B cells, dendritic cells, macrophages, and Langerhans cells are all antigen-presenting cells. And if they can't express MHC class II, they can't do their job. Now the T cells, on the other hand, although they use MHC class II, specifically the CD4 T cells, they're not dysfunctional. If you were to take these T cells and you were to put them in somebody else that had functioning MHC class II, they would work just fine. They would do their job. All the rest of these cells would not. They would not be able to, no matter what you did with them, if they can't express MHC class II, they can't do their job. Hence, the answer is E, T cells. Now, T cell activation is a two-step process. It requires 2 signals. It's another level of control, going back to the idea that if these cells are the generals behind the entire immune response, you really want to keep them in check. You want to make sure that they only get activated when you want them activated. Otherwise, they could run amok. They could cause all kinds of problems. So, we have a 2-signal theory. Signal 1 is the one that we've been talking about. When T cells are activated by APCs, the first signal that they have to receive is that signal that's coming through the MHC complex. So it's the MHC that's holding the peptide, hooks up with the TCR-CD3 complex, and we look for that match. Is the peptide specific, and is it the right MHC? That's the first signal that has to come through. Signal 2 is an interaction of APC co-stimulatory molecules with receptors on the T cell, specifically CD28 receptors on the surface of the T cells, and B7 or CD80 or CD86 molecules on the APCs. We include all of the nomenclature in there because they're both still used. B7 is an older nomenclature. The new nomenclature says that it's either CD80 or CD86. So one of those molecules expressed on the APC has to come in contact with the CD28 molecule expressed on the T cell, and that's the second signal. And only when we get both of those signals will the T cell become activated. Then, interestingly, the CD4 T helper cell can bidirectionally co-stimulate the APC. If it's a macrophage or a B cell, the T cell can actually send signals back to the APC to activate it. And we do this through another surface receptor called CD40 ligand on the T cell that binds to CD40 on the surface of the APC. So if you want to see a picture of this, here's the whole process, kind of what we just described here. We have a T helper cell, a naive T helper cell, being presented antigen by this B cell. Don't worry about the Th2 at this point. We'll talk about that in a little while. We have the B cell. It's processed this antigen, and now it's presenting a piece of it in class II MHC here on the surface. The class II MHC and the TCR have to bind together. The TCR is looking at that specificity. That's signal 1. The other part of signal 1 is the CD4 coming over and recognizing that this is a class II MHC molecule. That's all part of signal 1. That has to happen in order for the T cell to become activated. The other thing that has to happen is this interaction that's right above that, the CD28 and the CD80. That interaction is signal number 2. That's called the co-stimulatory molecule interaction. Both of those have to take place in order for this T cell to become activated. Now, once this T helper cell becomes activated, it's going to upregulate certain genes. It's going to upregulate cytokine genes. It's going to upregulate surface receptors as well. One of those surface receptors is CD40 ligand. The CD40 ligand from the T helper cell will then bind to the CD40 that's being expressed on the surface of the B cell. That bidirectionally stimulates, what we just talked about, bidirectionally stimulates the B cell to now become active. That B cell will then in turn upregulate cytokine receptors on its surface. Those receptors can then bind to the cytokines that the T helper cell is making. In this case, because it's a Th2 cell, it's making cytokines IL-4, IL-5, and IL-6. So those cytokines will then bind to the cytokine receptors on the B cell, and that promotes the B cell differentiation and activation that we talked about. That promotes the somatic hypermutation. That promotes the class switching. That promotes the memory cell response. So once the cytokines get involved, now that B cell starts to clonally expand. It starts to make memory cells. It starts to make plasma cells. And it starts to class switch so that as it continues to expand, it won't make IgM plasma cells, but will make IgG or IgA plasma cells, for example. So again, kind of going through step by step for that last slide, just to make sure we're all clear. MHC class II and the peptide interact with the TCR complex on the CD4 T cell. That's signal 1. CD80 or 86 binds to CD28. That's signal 2. These interactions activate that T helper cell, which then upregulates certain genes. The CD40 ligand on the T cell interacts with the CD40 on the B cell. That helps the B cell upregulate cytokine receptors. Also, the CD4 T cell makes and secretes cytokines. In this case, it's Th2 cytokines, IL-4, IL-5, and IL-6. These cytokines lead to the activation and proliferation of the B cell and are required for different processes in the B cell, such as somatic hypermutation, class switching, and memory and plasma cell differentiation. So here we have a question. A 6-year-old boy is being evaluated by a pediatric specialist after his primary pediatrician admits him to the hospital for his 3rd bout of pneumonia in a year. Specific serum tests reveal the presence of low-level IgM and no IgG or IgA antibodies. T cell levels and responses are normal. This patient most likely has a dysfunction in the expression of which of the following molecules? Is it A, CD3, B, CD19, C, MHC class II, D, CD40, or E, beta-2 microglobulin? The answer is D, CD40. From the clues in the question, we see that this young man has an immunodeficiency. He's got all kinds of recurrent infections with this pneumonia. He's got low-level IgM, so he has B cells. He can make antibody, but no IgG or IgA. That means he can't class switch. If you can make antibody but can't class switch, the defect is in that bidirectional stimulation, that CD40 ligand interaction, which upregulates the cytokine receptors. And again, because we see that in the question the T cell levels and responses are normal, we can therefore assume or deduce that the problem is not in cytokine production, but the cytokine receptor production. Here we have another slide of activation. This time it's not a B cell, it's an APC or a macrophage. And this time we're using a Th1 cell instead of a Th2 cell. So we see again the interactions are the same. Class II presenting that molecule in the context to T cell receptor. And then the CD4 coming over and recognizing that this is indeed class II MHC, signal 1 there. CD28 and CD80 binding together, that's signal 2. That signals to the T helper cell that it's got to get activated. Now T helper cells like to expand just like B cells like to expand. And the mechanism that they do this is shown here. They'll upregulate as part of their processes both secretion of IL-2 and expression of CD25 or the IL-2 receptor on the surface of the cell. So it's secreting IL-2 and it's expressing the IL-2 receptor. So it's going to self-stimulate. What do we call that? Autocrine stimulation, right? So it self-stimulates and we get expansion of these T helper cells. At the same time, it's expressing CD40 ligands, just like the last time we saw, that will combine or bind to the CD40 ligand, or excuse me, the CD40 on the surface of the APC. So CD40 ligand on the T cell, CD40 on the APC will then activate the APC, in this case the macrophage, so that it enhances phagocytic abilities. A couple of other notes in this slide. We see that we have another type of molecule being bound, the CD2 on the T cell, the LFA3 on the APC. That's an adhesion molecule. If you remember back when we talked about adhesion, those are adhesion molecules, and those can sometimes help with allowing these cells to bind to each other. A couple of other things on this slide. We see that the T helper 1 cell is expressing interferon gamma. That's another one of the cytokines the Th1 cells produce. Again, we'll talk about Th1 cells a little later. And notice that the APC, that macrophage, is producing cytokines by itself, IL-1, IL-6, and IL-12. Those cytokines are all involved also in differentiation of these CD4 cells into T helper 1 or T helper 2. We'll get to that in a little while, but just to be complete, just so that you see everything that's going on in the slide. Those are cytokines being produced by both sides. So, in text form, what we just saw, MHC class II peptide interacts with TCR complex on the CD4 T cell, and CD80 or 86 binds to CD28. Both of those signals required to then activate the CD4 T cell. The CD4 T cell can upregulate CD40 ligand, which will activate the macrophage and enhance phagocytosis. It can make and secrete cytokines such as IL-2, and it will also upregulate its expression of CD25, the IL-2 receptor. This causes an autocrine proliferative signal. Th1 cytokines that are being produced at this point include interferon gamma. These cytokines lead to activation and proliferation of cell-mediated responses. So T helper 1 cells are involved in cell-mediated responses. CD8 T cells, NK cells, phagocytes, all activated. Here we have CD8 T cell activation. It's pretty straightforward. Once you get the CD4 T cell activation, we have the signal 1 and signal 2 going through, activating this pre-CTL to upregulate IL-2 and the cytokine receptor, just like we saw last time. We also have cytokine influence here, interferon gamma that can come from macrophages or come from other T cells, and TNF-beta as well, that can activate that CTL to become a fully functional, enhanced, activated cytotoxic killer cell. But again, the process is the same. Have to have both signals. Actual form here, pretty much the same thing. We get signal 1 from the MHC through the TCR. We get signal 2 from CD80 binding to CD28. Then we get activation of the CD8 T cell. It will autocrine stimulate through this IL-2 cell-stimulating pathway. Other cytokines, such as interferon gamma and TNF-beta, can influence the activation status of the CD8 positive T cells, and these cytokines lead to activation and proliferation of these cells, which will then go and affect their cytotoxic functions. So a question here, a T cell researcher is studying activation of memory T helper cells. He notes that incubation of T cells with their specific antigen does not activate these cells. However, when supernatant from a culture of activated T cells is mixed with the previous culture, the cells proliferate. Which of the following molecules was most likely in the supernatant? Is it A, MHC class II, B, MHC class I, C, CD80, D, CD28, or E, IL-2? The answer is E, IL-2. The key to this question is understanding that this is coming from a supernatant. Remember, supernatant is the media that these cells are resting in, or the cells are responding in. When you transfer supernatant from one culture to another, you transfer only secreted molecules. The cells themselves don't get transferred. So because of that fact, then the answer becomes clear. Class 2, Class 1, CD80, CD28, while all involved in stimulation, aren't secreted. They're all membrane bound. So the only one that could be affecting this change would be IL-2. Now that we have our T cells stimulated and activated, we have to figure out a way to get them to stop. So the T cells have embedded in them a mechanism to shut themselves off. Is that important? Absolutely. If you have a T cell that gets turned on and never turned off, then you would just have continual expansion, continual proliferation, and we would all be walking lymph nodes by now. So we have to figure out a way to shut these T cells off. And the mechanism by which we do this is this molecule called CTLA4, which you can see at the bottom of the slide. When a T cell gets activated, as part of the expression profile, remember it upregulates CD40 ligand and it can upregulate CD25, it can secrete cytokines, it also upregulates the expression of this molecule called CTLA4. The CTLA4 comes to the surface of that T helper cell and it will bind to CD80. Now this function serves two purposes. As it binds to the CD80, it can prevent CD28 from binding, thereby cutting off that co-stimulation signal that's required for the T cell to become activated. Secondly, what it can do is once CTLA4 binds to CD80, then it gives a signal. It sends a downstream signal to the T cell to become shut off. Now, we see the magic of how this all happens. Once the T cell shuts off, it stops expressing CTLA4. And again, CTLA4 is now downregulated off the surface of the cell. The CD28 is upregulated, and if that T cell needs to become stimulated again, it can. There's no CTLA4 blocking it. So the overall mechanism here is you get short bursts of T cell activation. So they'll turn on, and then they'll shut themselves off. That CTLA4 gets downregulated, and then it can turn itself on again. Once it turns itself on again, the CTLA4 gets re-expressed and can turn itself off. So we get short bursts of activation, and if the T cell needs to repeat that, it's fine, but at the end of the day, we have the T cell automatically shutting itself off, and that's critical in maintaining regulation of the immune system. Now, CD4 versus CD8, some of those points just to again emphasize. CD4s are your helper T cells. They make cytokines. That's their job. They can regulate the responses of CD8s, macrophages, NK cells, all of the cell-mediated responding cells. B cells must be activated also by these T helper cells before they can produce antibody. Now, CD8s on the other side, they're the cytotoxic T cells. They promote destruction. They must be in direct contact with the target, and they use molecules such as perforins and granzymes, which we'll see shortly, in order to affect this change. CD4 T cells, not cytotoxic. They make cytokines. CD8 T cells, cytotoxic kill cells. So we have here another question. A pathologist is seeking to diagnose a patient's disease after finding an abnormally high amount of mononuclear cells in a bone marrow smear. She has made a tentative diagnosis of lymphocytic leukemia based on cell morphology. A more definitive diagnosis can be made by flow cytometry. Antibodies against which of the following would be most effective in identifying these cells? Is it A CD3, B CD4, C CD5, D CD8, or E CD44? And the answer is A, CD3. Because we need a general diagnosis here and we're looking for lymphocytic leukemia, we want to pick the molecule that has the most coverage, that is the broadest. And out of the molecules that are available for us to choose, CD3, remember, is expressed on the surface of all T cells, and so it would be the smartest choice in going to look for and characterizing a lymphocytic leukemia.
This concludes our discussion on cell-mediated immunity. In the next section, we will talk about effector functions of the adaptive immune system.